And great. So as Doug pointed out, I'm coming at this basically from a sort of a skeptical transplant nephrologist point of view. And with respect to my disclosures, I'm a non-inventive contributor on the IP that's been discussed in this talk, developed in the course of a PhD, essentially, but I have no commercial relationships outside of that. And I'm going to try to avoid repeating some of the concepts that have been raised by prior speakers, but I do want to revisit this issue of our current strategy, because I personally don't feel, at least as a liquid biopsy biomarker, that donor-derived cell-free DNA is the magical silver bullet. But I think a key reason that it's seen such adoption, particularly in the US, is really just how woeful our existing non-invasive strategies for the detection of rejection are, following specifically kidney transplantation. And so there is a lot of rejection that goes missed. There's opportunity for early intervention that is missed, and creatinine as a marker of rejection has poor specificity. The other complexity, and I think this is something that we need to be careful about when assessing the performance of biomarkers like donor-derived cell-free DNA, is that kidney biopsy itself, which is our gold standard, has created basically this paradigm based around the Banff criteria, which is an interpretation of allograft injury. But whilst it is the gold standard and the prevalent sort of method for diagnosis and treatment of rejection, may not necessarily accurately reflect tissue injury. And so it could very well be, I'm certainly not suggesting this is the case now, but it could certainly be that donor-derived cell-free DNA or other biomarkers that are developed, in fact, provide further risk stratification about the significance of some of the rejection we're seeing under the microscope. And it could very well be, as many of us suspect, that some rejection that we're seeing or some inflammation that we're seeing under the microscope may in fact not be alloimmune, and or may in fact not be of any prognostic or treatment significance. So today, the focus of this session, at least, has been on the sort of the DNA biomarker area, but I guess I do recognize that there are also concurrent, quite significant investment in biomarker development elsewhere. And I think interpreting donor-derived cell-free DNA's space in clinical practice in the future probably also going to complement and add to, rather than entirely replace, the additional information offered by other multi-analyte platforms and technologies. So I'll skip a discussion around cell-free DNA because that's been well covered. But what hasn't been well covered is the contributions and the variation that can be seen in total cell-free DNA level. And in fact, before there was this focus on chimeric cell-free DNA in the context of transplantation, as well as concurrent with the developments of these assays, there has also been a wide variety of study just looking at total cell-free DNA in non-chimeric scenarios. So in people that are not transplant recipients, basically. And what is apparent is that there are many conditions which modulate total cell-free DNA. Critical illness is associated with significant increases in total cell-free DNA, as are infection and a variety of physiological states, including things like exercise. And so cell-free DNA is not something that's unique to transplantation. It's something that's present in everyone. Its exact role is uncertain, but what is clear is that it's highly variable in physiology and in pathology. And this is important, I guess, in understanding potential confounders to the value of donor-derived cell-free DNA measures, particularly those that are reflected as a proportion of total cell-free DNA. So we've already discussed basically this concept of donor-derived cell-free DNA. I tend to call it graft-derived cell-free DNA because it comes from the graft, and in most kidney transplantation, the donor is deceased, at least in Australia, and no longer really a part of this world. So occasionally you'll see captions that say graft-derived cell-free DNA, but that's synonymous with donor-derived cell-free DNA. And so basically what we're talking about is chimerism, assay methodologies applied to cell-free DNA. And I also got to catch a little bit of the prior session talking about chimerism analysis of genomic DNA from circulating cells. And this is just a different analyte, basically. And so many people have looked and developed assays targeting all of the common sources of genetic variation. And the methodology I'm proposing today exploits ubiquitous copy number variation. And what's so lovely about copy number variation, not only does it account for a large number of base variation between individuals, but we can fit the entire digital PCR assay within the copy number variation. And so in scenarios where there is a homozygous copy number deletion or a zero copy number, there is no assay binding, there is no amplification. And so we create a negative background against which quantification is performed using digital droplet PCR, which allows for absolute quantification. So we're just going to talk a little bit about that in more detail. So you will all be aware that copy number variation describes these large sections of DNA. So in the case of this assay, the majority of the CNV we are targeting are between sort of 1,000 to 5,000 base pairs, and they may be present in variable copy number between individuals in the population. And so what we are interested in are combinations of copy number genotypes in which the donor is 1 or 2 copy for a particular locus and the recipient is 0 copy, or vice versa, to provide informativity for the recipient. And so to maximize the probability of this occurring, we took a population genomics approach, and we basically identified copy number variation loci that have a 0 copy prevalence of approximately 50% in the population, a 1 copy prevalence of approximately 40% in the population, and then are very uncommonly or uncommonly 2 copy and very uncommonly 3 copy. And so this maximizes the probability that any particular unrelated donor recipient pair is 1 copy at one locus and 0 copy at the other across the 2 genotypes. And so we conducted modeling which basically assured informativity in unrelated pairs with multiple informative markers being available for assay, and also performed similar modeling in related pairs for living donor transplantation. And we settled on an assay panel of about 31 markers, which, as we've seen in both allogeneic hematopoietic cell transplantation, but also in a cohort of kidney transplant recipients, is more than sufficient informativity for clinical utility. And so the underlying quantification relies upon a basic sort of TACMAN probe chemistry. So the idea is that there are forward and reverse primers and a TACMAN probe or related sort of concept probe that are specific to the CNV locus. So they do not bind anywhere else in the human genome. And so if that CNV locus is present as a 0 copy, no amplification and generation of fluorescence occurs. If it's present in 1 copy, amplification and fluorescence is produced with PCR product. And if it's present in 2 copy, roughly double the concentration is produced. And so this basic digital PCR platform thus allows absolute quantification. And so I'm not going to talk about that because it's already been discussed earlier in the piece. So this is a sort of I'm going to talk through basically what we see and the assumptions we make. So this is basically each bar here represents one assay, one primer probe combination targeting a specific CNV locus. And this is the assay panel or a part of it, run on cfDNA extracted from the plasma of a transplant recipient. And so this is what we see. And basically, you can see with the eye of faith, a clumping of markers up. They're sorted basically by concentration. And you can see a clumping of markers here, a clumping of markers here. And then what you can't see, because the concentrations are so low, is a clumping of positive markers down here that are at very low concentrations. And then there's a whole bunch of negative markers off to the right. I hope you can see my mouse cursor. Let me know if you can't. What happens then if we were to extract genomic DNA from the buffy coat of this transplant recipient and run the same assay panel? And what we can see here is that this clump of markers here on cell-free DNA, the same assays are present as a clump of markers here in the recipient's genome, this clump of assays here, likewise in the recipient's genome, and then nothing for any of the other markers. And so basically, these are 2 copy, these are 1 copy, and these are 0 copy. And so if we were to zoom in then on the markers that are absent from the recipient's genome, what do we see? Well, we see markers that are absent from the recipient genome up here, and we see some detectable sequences present in the plasma of the transplant recipient, which are not present in the recipient's genome. And so what would happen then if we went and we retrieved DNA that was stored at the tissue typing laboratory for the donor and ran this panel against the donor's DNA? And what we can see here in the donor genotype is 2 copy markers that correspond with the higher concentration graft markers in the chimeric sample, and 1 copy markers which correspond with the lower concentration markers. And then in those cases where there are no detected target, those are both absent in the recipient and the donor genome. And so what we do basically is that we take the median of those readings as the graft-derived cell-free DNA or the donor-derived cell-free DNA concentration, and then we take the median of the recipient 1 copy markers as a measure of total cell-free DNA. And so this particular digital PCR-based methodology allows measurement of the graft-derived cell-free DNA somewhat uniquely as a direct absolute quantification, copies per mil. We also measure the total cell-free DNA, and then we can also calculate the proportion that is donor-derived, the percentage donor-derived cell-free DNA, which many other sequencing-based methodologies can only report as the first outcome because they require pre-amplification as part of the sequencing methodology. And so this is another example sample. So this is cell-free DNA extracted. These are all recipient 1 copy markers. These are recipient 2 copy markers. And here's the graft-derived cell-free DNA in low concentration. And you'll see there is a little bit of variation in the results. These are reflective of the donor genotype. So 2 copy and 1 copy, which we don't distinguish between. We just use the median value because stochastically that median value is most likely to be a 1 copy measure. And these are multiple independent assessments which you as the analyzing sort of clinician can see as part of the interpretation of the calculated donor-derived cell-free DNA quantity. And so we ran this assay methodology in a sort of a diagnostic validation study in Australia, looking at adult kidney transplant recipients. And we were looking at the correlation between these independent measures, graft, total, and percentage, and correlating those against the results of biopsies that were done in proximity to those measures, and looking at the diagnostic performance essentially of those measures. And so these were people post-transplant who had evidence of graft dysfunction, which in the majority of cases was a rise in creatinine that led to the biopsy. And so unlike other approaches, we have looked at each of the measures. So on this left panel here, is the absolute measure of donor-derived cell-free DNA in copies per mil. And on the right panel here is the, let me move this out of the way, is the graft fraction. So the donor-derived cell-free DNA as a percentage. And then in the middle here is the total cell-free DNA concentration. And I guess one point to make here that I sort of thought was interesting as a clinician is that many of the people that did not have rejection but had graft dysfunction, or not many, but some of them, had very high levels of total cell-free DNA. And it emerged that these are people who actually had one person had urosepsis as a result of the biopsy. Someone else had horrific CMV viremia. So a good illustration of how concurrent illness, including sepsis and infections, can modulate the total cell-free DNA. So you could imagine if this was an individual that had rejection and sepsis, the rise in total cell-free DNA, which can be quite profound, 4 times the average, would reduce the sensitivity of the graft fraction as a percent, or could reduce the sensitivity of the graft fraction of a percent, because the denominator of that measure has been increased significantly. But anyway, to cut to the chase, we basically saw significant increases in the graft-derived cell-free DNA or donor-derived cell-free DNA in absolute terms, as well as in relative terms, primarily for antibody-mediated rejection, not so much for cell-mediated rejection, and particularly not for borderline cell-mediated rejection. And I think if you were to look across the studies, particularly borderline cell-mediated rejection, the donor-derived cell-free DNA does not perform particularly well for, and cell-mediated rejection, particularly milder forms of cell-mediated rejection like type 1A cell-mediated rejection, are somewhat controversially diagnosed as a result of donor-derived cell-free DNA. So really, I think that from my perspective, the signal is very strong for AMR. And also uniquely, we looked at the performance of each of these measures as it related to the specific histological lesion. So this is the BAMF score, and each one of these little abbreviations describes a particular type of inflammation or damage observed on under the microscope by the reporting pathologist. And it is this scoring system, this semi-quantitative scoring system, that is essentially used to make a diagnosis of rejection based on biopsy. And what we found was using the graft-derived cell-free DNA as an absolute measure was far more specific in the lesions. So each of the lesions where there was a statistically significant association with the scoring, the semi-quantitative scoring, is circled in red, basically. And we found that absolute measures correlated or were most specific to antibody-mediated microvascular inflammation type of lesions, whereas the relative measure of graft fraction was actually less specific to those, and also increased in other more chronic sort of scarring processes. And so at least in this paper, which was published in AJT, we argued that in fact one might take the approach that in performing an assessment, a donor-derived cell-free DNA assessment, you could review each of these results: the absolute measure of donor-derived cell-free DNA, the total cell-free DNA, and the fraction, to try to sort of understand the unique patient circumstance in more detail. And so this is looking at antibody-mediated rejection lumped together, so acute and chronic active. And so what we can see here is that absolute measures of graft-derived cell-free DNA for both acute as well as chronic active ABMR were higher compared to all other pathologies, and then the same with the relative measure of graft fraction. And at least as it related to ABMR, the sort of the box plot discrimination seemed a little better. But if we look at the ROC AUCs, they were both good. So this is for acute antibody-mediated rejection, and this is for both acute and chronic antibody-mediated rejection. I did not include CMR in this because we didn't see a signal for CMR, so we didn't lump everything together and call it acute rejection. It would have still looked pretty good, but we didn't feel we had sufficient sample to sort of ethically do that. And so we can see here, compared to creatinine, which performed woefully for both classifications, both the graft derived as an absolute measure and the graft fraction as a relative measure performed pretty well. And so this is basically the breakdown of diagnostic performance. So we defined our thresholds for the absolute measure at 21 copies per mil, and for the relative measure at 0.75%, and for acute antibody-mediated rejection, pretty good. And then for the composite endpoint of all antibody-mediated rejection, also pretty good, particularly if you compare it to our existing paradigm. I think the one point, and I think to highlight the points that were made previously, the negative predictive value is really the star here. What is not the star? This is an all-comers group, so we use no modification like DSA, for example. The PPV, and this is not just us, this is everyone that studies this, the PPV is disappointing for donor-derived cell-free DNA in general, but it's the negative predictive value that's the real star. And the diagnostic odds ratio, which is a measure of diagnostic utility, is also very good for this assay approach. And so just to really highlight, I mean, other speakers have done this already. There are a number of other methods. What is unique about this method, and it's only shared with one other approach to the best of my knowledge, is the employment of digital droplet PCR and also the primary quantification sort of class of absolute quantification as opposed to relative quantification. And so I thought, just to highlight my original point around this issue of liquid biopsy, if we take the best AUCs and the best sort of sensitivity and specificity performances from the literature, and we think about how this might affect someone who's got graft dysfunction, if it was increased, then you would certainly be concerned about rejection. You'd probably do a biopsy to figure out what type of rejection, cellular or antibody, because you'd need that to determine the correct course of treatment and to do some prognostication. If it was negative, well, you've still got graft dysfunction that's unexplained, and it would certainly dramatically reduce the likelihood of rejection, that's for sure. And so I would imagine, for example, in a pediatric cohort where biopsy is certainly far more complex undertaking, you may be reassured by that and you may choose to hold off. But certainly in an adult population, if the creatinine's up and you don't have an explanation for it, even if the test is negative, you're probably still going to do a biopsy. And there are many reasons for that. There may be other pathologies that are affecting the graft that have not caused a rise in the donor-derived cell-free DNA. And I think a classic example of that is borderline cell-mediated rejection, because we do, or at least I as a clinician, do treat some borderline cell-mediated rejection. And so if the test can't reliably exclude that, we're still doing a biopsy. Where I think the value of this test really is in monitoring, particularly high-risk people, and also in monitoring response to treatment. So just to pull it all together now, what I've presented is a simple digital PCR approach that is very similar to digital PCR-based cellular chimerism tests, except the analyte is different. It's cell-free DNA, and this permits absolute, total, and relative quantification. No prior genotyping is required because of the assumptions I've communicated earlier, and in fact, we've validated those assumptions, and we've shown when you look at donor and recipient DNA, those assumptions hold true. From my perspective, donor-derived cell-free DNA, I think of it as a graft microvascular injury biomarker. I like to think of it a bit like a CRP. And I think if you're going to take that approach, you want a cheap and cheerful test, a simple, cheap test that you can do regularly and not dwell upon too much. I think it's got very good sensitivity for AMR and ABMR, and that high negative predictive value is great at excluding those things, but it unfortunately does not replace biopsy when other potential pathologies are at play, and particularly in an adult population where the risks of biopsy are generally pretty low. And I think that's where further work is required, because I think there may be role to combine this with other biomarkers, as well as combine it with other assessments of graft injury that may emerge over time. So we're going to be getting a lot of good clinical utility studies from very large prospective cohorts, I think, in the near future, which is going to be good. I think in the meantime, it's helpful to play to its strengths, which really relate to AMR. And I think this issue of urine cfDNA, I agree, is also a tantalizing one for further study. Thanks very much.